Okay, so I think maybe at the beginning we could just it'd be nice to get a sense of where everybody is on, I guess, the progress of the analysis. I think I was planning on just focusing on the post-processing steps, because I think I had already worked with you all to a certain extent on um, like the pre-processing. So it'd be good. I think that most of you have already gotten some like level of the traces, like from all the ROIs. And I think it's like a matter of now taking those traces and merging them with behavioral data to get some sort of event-related visualization. So let's see. I think I have some notes on where everybody is.
Is everyone using Inscopix or?
No, yeah. So I think you're let's see, yeah, you're the only one using the mini scopes. But I think that the analysis like afterwards is going to be quite similar, because everyone's going to be working with basically an array where you have ROIs on the y-axis and then time on the x-axis, and then fluorescence is like the activity. Okay, so I have here that Callie, we had concatenated some of the data, the video data, and we ran it through Sweet2P, and then we made some code to push those data through that Sweet2P output through the event-related analysis, and that I think we need to do clustering next.
I think so. I think we had just gotten to the point where we're trying to figure out the timing of like when the stimuli were happening to see the responses.
Okay. Yeah, I think I remember that. We had just like entered in the timestamps, right? Just like, yeah.
And then I think we ran into issues of how I had lots and lots of cells, and that was causing an issue trying to make like 100 graphs.
Yeah, I mean, I think it's fine because we can. I'll explain some of that later for everybody as well. But yeah, I think there's a simple way to fix that and prevent that that bottleneck from happening. And then Jennifer and Brandon, remember we ran the data through the my pipeline, but I know you all were working on getting Sweet2P up and running. I'm curious to see how Sweet2P performs for y'all, because if it seems as if the signal to noise in your data are high enough, and the cell detection is adequate, then it might be worth just sticking with Sweet2P. So just wondering what your progress is.
So I haven't made a lot of progress. I'll just tell you, it's pretty much all starting now. So I mean, I have it all installed, but actually running my data through it, I haven't done yet. So, and I think Brandon has it installed now too. I mean, it was a pretty easy installation for me.
Yeah.
So yeah, and it seems like, I mean, the GUI is very clear. I don't think it'll be very hard, but yeah, I haven't had time set aside for this until now.
Okay, sure. Yeah. I guess what we could do is, let's see, since Sweet2P might not be applicable to everybody, if there's time at the end, I could kind of give a rough walkthrough of how to use Sweet2P.
I mean, that's totally fine. I think I would really like more of a tutorial on SEMA anyways. So, because there's a lot available online for Sweet2P, right, tutorial wise.
Yeah.
Yeah.
Yeah. Okay, sounds good. Okay, and then Victoria, you have Inscopix data. I think I know like what the structure of the output data come from or are in coming out of the pre-processing pipeline, but be good to just make sure. And I think now we just need to kind of if the format of all this data are fine and they can be like thrown directly into the post-processing pipeline, that's great. If not, then I guess we need to like just write up a few other lines of code to convert the data and pump them through. Okay, so let me share my screen. Are you seeing a GitHub page right now? Okay.
Yeah.
Cool. So I will be mainly talking about not this repository, but this one. It's kind of more bare bones than the pre-processing pipeline. Some of the documentation and stuff are still being worked on, but there are several codes that I think can be relevant to you all. I sent the link to it in Slack, and I think the idea is we can try to have you all like kind of follow along. Basically creating a new Anaconda environment and actually running some of the data through. So I have some sample data similar to the pre-processing pipeline that's in NAPECA post, and then sample data. It's one of VJ Nambudieri's 2-photon calcium imaging data. So these are like files that directly come out of the pre-processing pipeline. And I'll show you how those data look, just so that for those of you who aren't using, I guess, the NAPECA pre-processing pipeline, you can get familiar with how these data look like, and we can work together to adapt your data to fit the scheme for loading the data successfully in. Okay, so I think we can start out with, is anyone going to follow along? Do they have like a computer that we can install? If you don't already have it, install Anaconda prompt, as well as run some Anaconda prompts to install an environment, and then the requirements.
Yeah, I'll download it right now. I don't have it at the moment, but I can do it really quick.
Okay, and yeah, we can see if there's some time for you to catch up. If not, this is recorded, so.
Oh, does Anaconda use Python?
Yeah. Well,
Could
Anaconda.
we use Colab instead, or only Anaconda?
I don't really have everything set up for Colab, unfortunately.
Okay, that's okay. I'm downloading Anaconda.
Okay. So you don't need me to go to like anaconda.com or anything like that. Have you installed Anaconda before? It's pretty straightforward, I feel like.
Yeah, it's downloading.
Okay, good. Okay. Let's see. Okay, so then I have to do it myself right now. So I would say what you all could do, let's see, if you already have GitHub ready, you could just fork or clone this this repository. Otherwise, just to make it simple for you all who don't, you could just simply download this as a zip. And so that's just pulling all this code from this repository and downloading it as a file. So one thing to note about this method is that any like additions, or new commits, new updates to the code won't be reflected in this local kind of saved file if you just download the zip. Let's see, I think we already have this on this computer. So I'll just open up where that is.
Sorry, how do you download it as a zip file?
So you go to this link, you go to code, oops, and then download zip.
Are we in the sample data file?
Oh, sorry, yeah, I kind of I should have mentioned that we have to be, yeah, you have to be in like
Oh, I see.
the root kind of web page.
I see. Okay, got it.
Yeah. Okay, so yeah, I'll wait a little bit for that to download. It should be pretty quick. But here I'm just navigating to that. Yeah, I guess we could use this one. I'm just navigating to that repository. Remember, I just unzipped it, and now we basically have all those files that's on the repository. So this requirements.txt is going to be important in a second. We're going to install all of these packages that are important for the analysis. And then you have all your code here, as well as the sample data. Do you need a few more seconds, Victoria?
I haven't downloaded. Now it's asking kind of which application, like Jupyter, Lab, Notebook.
Are you talking about this zip file?
No, the Anaconda.
Oh, okay. Say what's
The
the
zip file I
question?
have. I opened up Anaconda and it's Anaconda Navigator.
Anaconda prompt. So once you have it installed, you should type in the Finder Anaconda. So Navigator is kind of the GUI-based, like visualized version of Anaconda prompt. Yeah, I guess you could use either, but I guess I'm more used to Anaconda prompt at this time. And it doesn't really hurt too much to get your feet wet a little bit with the command prompt format of executing lines of code.
I don't know where the Anaconda prompt is. Maybe I downloaded the wrong thing.
Well, I guess you could use Anaconda Navigator too. So you can open up Navigator as well.
Mm-hmm.
Okay. Does anybody else need help with installing Anaconda and stuff, or do you all already have it installed?
I have it on a different lab computer than the one I'm currently on, but I'll just like watch and follow along and look back
Okay.
at the recording.
So this is still a little bit helpful, I guess.
And I mean, I'm using the terminal window in Visual Studio Code,
Oh, okay.
the Anaconda prompt, but it's
That does the same thing.
Yeah.
Okay, cool. Is it opening up for me? Okay, there it is. So yeah, maybe I'll just jump back and forth. So does this, yeah?
Just a quick question. So for this post-processing section, so I have like a SEMA environment and I have a Suite 2P environment. Like, do you recommend creating another environment?
Yes.
For the post-processing?
Yes. So
Okay.
that's what we're doing right
Okay.
now.
That's what I thought. I just wanted to be sure. Okay.
Yeah. Thanks for clarifying.
Thanks.
Are you all at this point? Okay, cool. So, admittedly, I haven't really done this recently, so I'm hoping that this works. But you should go to the environment tab. This will list like all the environments that you have, I guess, installed and created on this computer. Just briefly, environments just a specific environment, like a single instance of a set of packages for Python to load in, and you can have multiple different environments that have like different mixed and matched packages that you want to use for a specific project. The reason why you have different ones is because different projects might require different packages. So I think what we're going to need to do is click the import button and then specify a name. Let's just call this post navigator. You don't need to put the navigator. I'm just doing it because I think I'm going to make something similar through the Anaconda prompt. And then we're going to go and find that. Oh, this is in. This might not actually work because this is a requirements.txt. I think what we're gonna have to do for you, Victoria, is.
I don't mean to flip everything on.
Yeah, no problem. I think it's still good to. It's still good to work through this. So how about this? I'll just show you kind of the workflow of this, because it might take a little bit of time for you to do this.
Sounds good. Yeah.
Is it? So you couldn't find the Anaconda prompt at all?
No, but I will find it later.
Okay, yeah, no problem.
I just don't want to take any more.
Yeah. So what you should do is do create. And make sure that it's a Python 3. It's going to create this. Oh, I have too many folders now open. So while that's being made, you're going to want to go back to this that folder that contains all the repository and open up this requirements.txt. And so you're going to go through, so this is like a window showing all the possible packages that you can install. You're going to want to type in each of these besides Python. Let's see, let's do scikit-learn, or I can just do this. No, I don't need to. It really wants me to update. You should do not installed. Yeah, so then just like search for the appropriate, here it is, package. And then I'm just trying to see where you can get the correct version, but I think just install whatever is there, and then hopefully the the version is fine. So then just go down that list and install these. Okay. So for everybody else, let's go back to Anaconda prompt. And then, so we're going to do the same thing to create an environment. We could do conda create. And then this is just like the name argument. So you're going to want to do -n. And then I'm just going to call it NAPECA post. And then Python is equal to 3.7. Let me know if anyone needs to catch up to this point.
Charles, can I just do like conda create and then the name prompt and the name and then have it reference the requirements text file?
I'm not sure if that will work. You could try that out.
Is it -f? Is that the conda prompt
You
for
could try that. Let's
finding
do
that file?
Yeah, so that I know works if it's a YML file. That's like just another like conda like specific requirements file. But let's see if this will work. It might actually work.
Well, I'm doing it now, so we'll see.
Oh, is it working? Okay. So let's try.
It said it failed.
Oh, well,
Yeah.
I'm curious still.
And I'm pretty sure I was in the right directory, so.
Oh yeah, I'm not even in the right directory right here. Yeah. This might actually just make this environment, but just to fail at the requirements. Did it make it? Okay, cool. Not yet. So actually, I skipped a step. So what we're gonna need to do is in the conda prompt, we need to go to this, I guess, CD or navigate to this directory in the prompt so that we can directly reference this requirements.txt. So now you can see my, I guess, where I'm working in, the current working directory in Anaconda prompt is this folder. I'm going to go back and do this. Yeah. Yeah, I don't think it's liking that right now. But it's just going to be like another line that we'll use to install these requirements. I can make a YML file so that it's just one step to create the environment and install the packages as well in the future. Right. And then Python is equal to 3.7. And then you want to say yes here. Okay, let me know. I guess I'll wait here for a second just to make sure everyone's got to this point. I guess you can either tell me or raise your hand.
Can you scroll back up just a little bit so I can see your what you put in the window?
Oh, this one here?
Yeah.
I don't know why Anaconda Navigate is freaking out on me so much. Anybody need any more time? Okay. I just slacked you all. This is kind of more meta, like big picture, but this is just a document that gives you all the different resources that are related to these like pre-processing, post-processing pipelines. So this is actually a walkthrough of how to use the pre-processing pipeline GUI, and then the necessary scripts that we'll be talking about for post-processing. Okay, let's see, where are we here? Okay, so now that we have the environment created, we want to activate it. So let's just kind of activate whatever you called it. Here, I just make the post prompt. And so now you can see that we've activated this environment, and any packages, we can install specific packages into this environment, and any packages that are installed there, we can use that and reference that, load them up into our scripts. Okay, so then now we want to install that those packages from the requirements.txt. So we want to do pip install -r. -r is important to be able to tell, I guess, the pip to go into the actual requirements file. So you want to do this, and then okay, so there's something that you need to do here. So we need to go back into this requirements.txt and just delete Python. That's seemed to be an update, the recent update that that was made to this requirements that is actually not needed. So let's try that again. I think this should work. Yeah. So yeah, this is just now all the different steps for installing, or it's running through all the installation steps for installing all these packages. So let's see. So sklearn is good for like machine learning, functionality, stats model for stats, NumPy for just array manipulation, Seaborn Matplotlib for visualization, Pandas for Excel like sheets and data frames, and then the H5Py is for loading in the raw data. So if everything went through, then you should have gotten to this step. And now we can actually start to work with the code. Does anyone need any more time? This is going to be kind of the next transition into talking about the code now. Were you able to install everything, Victoria?
Yeah, I think everything's
Okay, well, we will see if the code works with these. I think you probably installed like the most updated versions of the package or the packages. Well, I mean, the thing is that some of the, I think, requirements that I have here, they're like older versions. So I'm not sure if they can be compatible, but we'll see. Okay.
On the Anaconda Navigator, do I open terminal? Open with
Oh, yeah. How do we do this? Kind of. Oh, I think I know how to do this. There should be not in the environments tab, but the one above it. It should, if you click it, it should show all like the different applications that you can open up. So open up Jupyter Notebook if you see that.
Yeah.
Yeah. And that should open up a new kind of window in like your browser.
Yes.
Okay, great. So now for everybody else, we want to do Jupyter Notebook like that, or I guess here you can launch it in the Jupyter Notebook. And Navigator is going to freak out on me. Okay, cool. So I'm gonna click that. It's gonna let me. Okay, good. So if you had already navigated to this folder in prompt, then it'll automatically open up its browser into that repository. So this will be kind of easy to just like go in and open up any of these notebooks. So the first code that we're going to talk about is this one. But just as a general kind of overview, all these codes, again, basically take in extracted signals from your ROIs, which is going to be typically an array again of ROIs by time, as well as behavioral events. The format of this I'll show you is in the form of a pickle file with containers that represent the different trial conditions. So let's say we'll be working with a session where the animals were administered on different trials across the whole session. Let's see, sucrose that was paired with a CS plus tone, and then no sucrose, which was paired with a CS minus tone. And then within those containers for each condition is going to be a list of time points when those events happened, and I can show you how to create those files. And yeah, so we're going to take those data, combine those two data types, and then create different visualizations in order for you to get a sense of how the data look in a general sense. And also there will be some plots that I think can be used for publication purposes as well. But I think for the most part, this is more of a set of tools for you to just be able to pump your data through and get a general sense of how activity looks relative to behavior, or some sort of like manipulation that you've done to the animal. So the first one will be this whole session event ticks. So if you click that, it'll open up. And I guess I'll just outline generally how these scripts are oriented. And I guess one kind of forward is that, yeah, all of these are going to be Jupyter notebooks. So maybe it's a little bit intimidating just seeing all this code. But hopefully I've formatted things in such a way that it's kind of easy to go in and change specific user set parameters, and then all you need to do is run all the cells, which I'll show you how to do. And the goal is that ultimately I'm having a few technicians, engineers working on turning this into like a GUI, so it's even more easier to run. So, and I need to add some more documentation, but hopefully this video is good enough. The format of most of these scripts is that I have just the importing block, and then I have this user-defined variables block, where for for the most part, I think you'll be working in like a block like this, where you can see there's specific variables that need to change. In this specific script, it's to you have to set the file name, just like the base file name, the directory where the data are located in, and I'll show you how to do that. The sampling rate of the data, and then typically there are some just like optional parameters that that you can like work with. Particularly here, this is like whether or not to load in neuroplug corrected data, which I don't think this might not be relevant to you, Victoria. And then the different conditions to analyze, and then how to normalize data. And then all this subsequent stuff is just like the guts of the analysis. And then typically there's some visualization pieces of code. So this particular code will allow you to basically visualize the activity of each cell across a whole session, and the timestamps of each event. At each timestamp, there will be these kind of dotted lines that indicate when they happen relative to the activity. And you can kind of scroll through. Well, actually, let's see. We should run all this first. So we want to come up here, and first we need to do a little bit of changing here, because this is actually just referencing my local directory, but where you've saved this this repository locally is probably different from where I have it. So what you're going to want to do is go back to this folder where you have all the you downloaded the repository, and then let's see. Well, actually, you need to go to here. We need to go to sample data. So the signals, the extracted signals are here, and the events are here. And I'll talk about how to, I guess, create these if you're going from Suite 2P, and how to adapt the Miniscope data to that as well. But first, let's just copy and paste this directory into here, the fdir, so that's just file directory. And then this is the root name of the file, which I have here. So you can either just like restart and run all, or just just click into the first block and do Shift Enter. Actually, I think there's something that you might run into an error, actually, because I see this package.
Yeah, I had to install Plotly.
Oh, okay.
Well, it's installing right now.
Oh, okay. Good that you identified that. So maybe I should show people how to do that. We actually need to get out of this to do that, unfortunately. So we'll have to, if you've made changes here, you can just save it here. I guess this is kind of good for me to show you, because hopefully it's not too confusing, but this might happen where you need to go back and install a requirement that that you don't already have. So we'll close out of this Jupyter notebook. Yeah, my computer is a little bit buggy today for some reason. Hopefully it just closed for you. I think if it doesn't, then we can just go back to here. Oh yeah, I think it's already out because this is not lit up. So I'm just going to do quit here and just close out. And then this should kind of go back to like that, but if it doesn't, you can do Control C. Is everyone here now? Back in the Anaconda prompt. And I think Victoria, you could just go back into the navigator.
Okay.
And, yeah. Okay, so I'm gonna do pip install Plotly. So Plotly is just a package for interactive visualization. Oh yeah, so for the Inscopics data, what's the format of the, I guess, the extracted traces?
Yeah, it's like a data frame. The first column is time, and then the subsequent columns are just the ROIs in there.
Okay, so then it should probably look something like this, right? Is it a CSV?
Yeah.
Okay, yeah. So then I think this should be just pretty straightforward like that, right?
Yes. Yeah,
Okay.
exactly like that. Yeah.
Cool. So there's going to be a little bit of adaptation, but it should be pretty simple that I can go over with you. Okay, so we're back here again. Let's open this back up. Hopefully this runs through. Let's see. Okay, it ran through for me. Let me know if you run into any of the errors. So I'm going to go through this. Just going to run through all these cells. And it should come up like this again. Let me know if you got to this step.
Real quick, I'm actually still trying to install Plotly because in the command prompt, it just never stopped. It's just on installing collected packages, and then there's no lines after it right now.
So it's like here, or you might want to like press enter, and because it sometimes just sticks. Like I think it's like it has.
Actually, yeah, that worked.
Okay, yeah. Sometimes prompt is also kind of buggy where it's like I think it's basically ready to finish, but you just need to push it a little bit.
Yeah.
Okay. Has anyone else gotten to this point? Did it go all the way through?
So just a quick question, which file was it? So when I put in the path, the file directory,
Yeah.
which file were we going for?
Oh, sorry. So you can keep the name, the file name as it is, because the file name is going to be similar between you and I.
I have that, but then like my directory.
Yeah, directory will be, so let's see, this is your extracted repository, right?
Right, but
And
is
then,
it like the neural pill corrected signals? Or
you're talking about this?
yes, which one did I want there?
It'll automatically pull this file as long as you
Okay.
have the right base name. But now that I'm thinking about it, I need to adapt this code a little bit if you're using Sweet2P because you won't, or Sweet2P or Inscopix pipeline because you might not have this specific file. So I have a script that I wrote with Callie to be able to create a file like this, to be able to load in here. So that's like coming, I guess, a step before all these scripts where essentially the data coming off of Inscopix recordings, as well as the data coming, or the Inscopix preprocessing pipeline, as well as the data coming through and out of the Sweet2P pipeline, they might be in a different kind of format than what like immediately this code wants. So I have another script that can kind of adapt that, which I wanted to get to like right after this. But for now, just wanted to make sure that this code works with the sample data. So as long as you are in the sample data directory, you can copy and then paste it here. I'm just going to run through again. So that's if you don't want to just shift enter everything, you can do restart and run all. Okay, so again, you can scroll through the different cells, the extracted traces, and then also, let's
I have a
see.
quick question.
Yeah.
On one of the cells, I'm getting a warning that there's no or multiple signal files detected using the first detected file.
Is that up here somewhere?
Yeah, it's in
Oh,
the
here.
block where, yeah, load time series data.
Oh, okay. Did you have everything pointing to the right place here?
So I'm in sample data. Let me see.
Do you have this folder?
Uh,
Like, I guess if you physically navigate to this.
I think so. Yeah, I think so.
And then just make sure that.
Oh, I got it.
Okay, nice.
That
Great.
was my mistake. Sorry. Okay.
Yeah, no problem. Okay, so hopefully everybody has this plot. Let me know, and feel free to just yell out or raise your hand if you need, like, if you're falling behind or anything. So I like this RY1 because when we zoom in, we can see that we have, so let me explain this a little bit more. So the red dotted lines are when the CS plus stimulus was presented, and then the blue dotted line is the CS minus. We can click each of these to like display or hide the different lines. And I like this example because if you like just look at the plus, which is kind of I think expected in this brain region. This is a OFC that whenever there was sucrose delivered, I think when the animal was licking and receiving the reward, there was a nice increase in activity. So I think this is a nice little tool to be able to look at how the activity of a cell evolves across the whole session, and how different events can relate to those activities. So here you can see that for the CS minus, there might be some in cases where there's some activity happening afterwards, but definitely like a lot lower magnitude than the sucrose responses or the CS plus responses. And you can take snapshots using this. Unfortunately, I think that you currently, at least, I mean, I've made this script a while back, so maybe the package Plotly has improved, have some updates. I'm not quite sure if you can export as a vectorized figure. But again, I think can be more used as a diagnostic. So a few things here that I wanted to mention. Currently, I have it just to set out to visualize 10 cells to show all of them, which I think there were like 60 of them here. And it might take some take up decent amount of like memory and stuff to load and everything. You can delete that and then or delete this 10 and the hashtag or the £. And then if you execute this all the way through, then it'll show all the cells. And see down here, it'll go through all the total number of cells. I just use 10 because it's a little bit faster to execute for now. Now yeah, go ahead.
This is like down at the very bottom of the code, but it's throwing back errors saying like, I think there's another package we're supposed to install as well, the Kaleido engine or Kaleido package.
That I am not familiar with. Can you spell it?
It's like kaleidoscope. So Kaleido.
Wonder if that's like for the. Can you screenshot that and send it to me? I haven't encountered that before. So you can't see this at all?
No, so I can see that just fine, but it's after like the whole session event.
Oh, this one?
Yeah.
Oh, okay. Let's see. Yeah, let's do that offline.
Yeah, it's not important right now.
Yeah, because this is to save, I guess, the figure. Yeah, actually this is kind of the workaround to be able to save this as a vectorized file. The only downside is that if you zoom in here, it's not going to like save the zoomed in version. It's just going to save like this zoomed out. But yeah, that's good to know that. I think that's kind of funny because this is a matplotlib package or part of matplotlib, I believe, and I guess for some reason it when it was installing dependencies, it didn't install that kaleidoscope one. Okay, let me note that down. Okay. So all this time you might be wondering, well, how, what format and how are we loading in the activity? So the loading happens here, and it's here. I'm trying to find the specific file, and then I'm using np.load to load it in. So right now, these data are in this np numpy file format. If you're using my pre-processing pipeline, it'll spit this out directly. But when it's loaded in, it's going to look like this. You don't necessarily need to follow along for this. Yeah, I don't know why my Python kernel keeps on dying. Oh.
Mine's doing that too, just saying that.
Yeah, that's kind of weird. Well, I've shown you how to get to that last step, so I don't think these you don't necessarily need to follow on for these next few ones. Is the whole Python Jupyter crashing? This is a little bit strange.
It's just fine in Visual Studio.
Huh. Okay. Well, hopefully it's just that one specific code. I'm not going to run it all the way through this time around. Let's see.
I mean, is Jupyter really sensitive to internet connection issues?
No, I don't think so, because it's not going through the internet. It's just using the one of your browsers just as a platform for loading it in. It's like all of the I guess loading and rendering of all this is just through your local Anaconda prompt. I don't think that it needs internet at all, actually.
Oh, I didn't know that. That's good to know.
Yeah.
Thanks.
Okay, cool. So I just ran it up to this load behavioral data block, but I just wanted to show you how the signals and everything look. So, I mean, it's pretty simple. It's just a NumPy array with like all the fluorescence values and then the shape of everything. So it's 61 cells by however many time points. So these are just like samples. So let's see, when do I want to show that? I will show how to get, so yeah, so Victoria, I'll work with you to adapt the Inscopix data, but shortly after this, I'll show people how to convert the Sweet2P data to appropriate format. And I guess long-term, I need to update this code in order to just automatically be able to detect the file type that you're loading in and automatically adapt those data to this pipeline. So this is signals, and then the behavioral data are here. So this is in a pickle format. This PKL is just another file format to save things. So NumPy is good for arrays, and then pickle, we can save data structures like dictionaries. So event frames is what gets loaded up ultimately. And so here you can see that we have a dictionary denoted by these brackets, and then dictionary is like a container, right? Where, or I guess similar to like just an analogy would be a file drawer where you have like the folders that you can put like documents in. And in this case, the dividers or the folders, they're kind of named after the behavioral events. And then within each of these keys, or what they're called, there's going to be a list or a array of time points of when the events happen. So here you can see we have all these different events happening throughout, and then also the time points. And see that these are in samples, so you're going to have to multiply your time points by your sampling rate. So how do we get those, create those? This is not, this code is not directly in that repository, because still need to polish it a little bit. But basically here, what we're doing is, again, specifying the the folder for where the Sweet2P data are. So this is like the root folder, because here this line basically figures out where the specific, let's see. Oh no, this specifically finds the Sweet2P folder. When you run Sweet2P, it'll make a Sweet2P folder where the output's going to be. So this is going to be the root directory of, I guess, where you have like the raw data, the H5 or the TIFF file that you load into Sweet2P. And then this is just like the root name. And if you run through like all these cells, basically what's going to happen is it's going to load in the fluorescence data that gets outputted by Sweet2P. This is this f.npy. And then it's just going to simply turn those data into a NumPy array. Oh, actually, there's a little bit more here. So when you go through Sweet2P, there's like that there's that step where you curate the cells, like tell it if indicate if a cell is a or an ROI is a cell or not. And so there's another file called isCell that gets outputted by Sweet2P, which is basically a list of booleans for each ROI that gets extracted here. And then here it's just saying that only the ROIs that you've deemed as a cell should be included in this NumPy array that gets saved into this extracted signals.npy. So then you would just grab that file and then put it into, I guess, the directory that you want to. Let me just close out. Into the directory that you want to process the post-processing data, or process those data through the to the visualization scripts. So, and then I think, let's see.
Charles, I hate to stop you right in the middle of this. Where did you get this file, the S2P conversion?
This one?
Yeah.
Yeah, so I'll send this over to you all. It's not in the repository yet because it's kind of specific to if you're using Sweet2P.
Okay, thank you.
And it needs a, I'll clean this up a little bit and like document it before I send it over, so it's a little bit more easier to use. Yeah, this is a file that Callie and I kind of wrote together. Let me think. So this next step is more related to people who don't have, I guess, don't have a way to extract specific timing information from their behavior. So I think the Jennifer, anybody who's using the Bruker microscope, there's going to be there's another script that you can run where it'll take the analog inputs and extract out the time points, the sample points from each behavioral event. So this might not be as relevant. But I think maybe for Victoria, since I'm not sure how you are saving your behavioral event timing, but one way you can create a file that's going to be compatible with these downstream analyses is doing this, where, let's see. So here we have you want to set the sampling rate. And then this is going to be a list of events for specific event type. So here I just have it set to stim. So if you have, let's see, I actually don't know what the exact manipulation you're doing. I guess maybe drug injection.
Yeah, I haven't done any yet, but I will.
Oh, okay.
Sure, we would do.
Well, let's say you're just doing like this might not be what you're going to do, but like if you are poking the animal at a specific time point, this could
Yeah.
be set as poke. And then let's say you had like 4 different or 3 different time points where you did that poke. You would just simply do, and you did it at 30 seconds, 60 seconds, and 90 seconds. You do 30 times FS. You don't really need this, 60, and then 90.
I see.
And then you could do that, make another one if you had a different, that's correct. And then different time points here. And then it'll basically save a pickle file that contains, that'll look kind of similar to, let's see, I guess I deleted it, but what that looks like our event frames.
And what is FS?
Oh, FS is the sampling rate.
I see.
Yeah, I'll comment this and document this a little bit better before I send it over.
Okay, sounds good. Thank you.
Okay, so yeah, that's kind of the one step that needs a little bit more development. I think these other scripts, this should run a little bit more smoothly. So the next script that you might want to run is this event-related analysis. I think this is the main kind of visualization code. So, and I have this a little bit better documented, all the like how to run this code, and then the different parameters that you might want to change in their descriptions. Again, just run that. This is just helper functions, and then the main code that you will be kind of adjusting is this again, this user-defined variables. So first off, let's just adjust the f name and the after again. So similar kind of situation here where we copy and paste the sample data. Okay. Let me see if this is, so all of this, all these parameters are already preset to work with these data. I think probably let's run through the code just to show you the visualization before we go into what each of these parameters do, because it might make more sense if we know how the plots look like. So just execute through all of these. These are just like loading in the data again and preparing some variables for the visualization. When you get to this, the start trial based pre-processing, I'll wait a little bit just to make sure that everyone gets to this point.
So many figures.
You got past it? Okay. That's good to know that everything's going or working. So before we get to the plotting of the figures, this this line of code is crucial. So I don't think you'll need to like go into like the guts of it, but it's calling this this function in the utils file to separate like a Python script in the repository. It's basically taking in the your signals, like the ROI by time signals across the whole session, and also taking in the timing of all the events, as well as a variable that you were going to set that dictates like the size of the window, time window around the event that you want to snip out, because these figures, subsequent figures, there will be event-related snippets of activity for each cell. So with all this information, it's going to go trial by trial and extract out just epochs of activity for each trial, each cell, and it's going to pump, stick it, or output this data dict file or variable. So let's go over to this first visualization step. So yeah, this was the step where I think Callie, if she's still here. Okay, she left, but we have this recording. She had like so many ROIs, and she had some issues plotting all this because it's just like hundreds of plots. But if you run into the situation where you have way too many cells, there's a way to just suppress like visualization here and just make it so that it just saves these plots as files that you can scroll through later. But hopefully this is not too intensive for your computers, but you can see here that now we get to these plots where each set of plots represents a single cell or ROI, and we have trial resolved responses relative to a certain event. So here we have the plus and the minus events. And then the activity is encoded in color. And then the trial average responses are shown here on the right, again, for each ROI. So you can see that this particular cell seemed to respond more strongly to the CS plus. And if you remember, when we were going through the whole session representation, indeed, this was that ROI1 where there was some decent time-locked CS plus activation. And then, I mean, so on and so forth. Okay, so let's go to the next few plots. So here, this is the trial averaged heat map where we have all the ROIs on the y-axis and then time again relative to that event on the x-axis. And yeah, all the trial average responses for each ROI are collapsed here. And there is an option to change the sorting. So if we wanted to, we could sort these ROIs based on the minus condition. So the order of the ROIs are the same between these two plots. And then second to last is the ROI average traces. I mean, this will eliminate a lot of like the variability across the cells, but I guess if you wanted a more population-based representation of the activity, you have that here. And then finally, just like brief quantification. You can set, or this is the time average quantification between the two conditions. And that's basically it for this code. But hopefully you can see that this kind of gives you a rough sense of how the population responds to different conditions at different levels of averaging. And just kind of briefly, we can go through some of these parameters that you might want to mess around with. Here, setting the sampling rate. This is going to be pretty important for visualization. If you get this wrong, then maybe the time scale will be wrong. If you ran my NAPECA preprocessing pipeline and set the sampling rate appropriately, there should be a just like a metadata file that contains the sampling rates that it'll pull that sampling rate from. So this, if the behavioral event, the conditions that you saved in the pickle file that I showed you here, or the one that's automatically output by the NAPECA pipeline, if you use that, then and the conditions that you want to analyze are, you want to analyze all those conditions, you can set this to none. But if you wanted to analyze some specific conditions, so let's say if you just wanted to look at the plus conditions, you can set that here. This is the trial windowing parameter. So you can see that our windowing was from -2 seconds to 8 seconds, and you can change that with this list here. It has to be a list, so the brackets and then the 2 entries. But yeah, you can set this to like -5 seconds to whatever. This variable is important for normalization. So some of those plots you saw had like z-score normalization. So this will be the end time point of the normalization window. So the z-score data were basically z-scored relative to a -2 to -0.2 seconds time window. You can basically, if you want to, just set this to 0 if you want. This is the event duration is just for visualization purposes. So you might have noticed that we have a little green line down here. That's just to indicate maybe to people reading the paper or the figure that your specific event happened for that duration. And then this event sort window is the window during which you will be sorting the ROIs by in the heat map, as well as the window to do the stats on at the end. I think this will be false for most people. This is more related to optogenetics during the recording. This is whether or not to z-score for the plots, and then parameters for ROI sorting, and then parameters for saving figures. Yeah, particular figures. I think that's mainly it for this. Does anyone have questions about this? Okay. Did everything run through for everybody? You're able to get to these plots? If people are still following along?
It ran for me.
Okay, great. So yeah, again, I think the key files that need to be loaded in are the event frames, as we saw before, as well as the signals. One thing that I wanted to note about this is for the signals, I think in this particular version, I have like a update that's going to be, I'm going to put out very soon. So I showed you like all these conversions, right, of the output from like the pre-processing pipelines. What I'm working on is a way to just input in Excel sheets or CSV files directly into this, so that you don't need to do some Python coding to convert. So the idea is, instead of needing to load in a NumPy file, you can just save an Excel sheet that's, let's say, ROIs by time into this. You just specify that in that above block of code for the user-defined variables. And for the behavioral events, I think we should discuss this to see what people prefer in terms of how they want the behavioral data to be oriented. I think different programs can have different like format of the behavioral data. So one idea is to have a CSV where the first column is the event type, and the second column is the time point that the event happens. And so again, that should simplify the loading in of the data. But we can talk about that after this next last code. So I'm going to quit out of this. Let's see, I think there's 2 more scripts, but I think this one might be of interest to a lot of people. So this is a code that, let's see, do we have everything? There might be other packages that need to be installed. This is a code that my an undergrad technician has been working a lot on to kind of optimize. This is a code to be able to take in the event-related data that we just ran through. So the prerequisite is indeed to run through the event-related analysis, because that spits out a file that contains all of the event-related snippets, and ultimately it takes those data and performs clustering, unsupervised clustering of those data in order to group ROIs or cells based on how they respond in different conditions. So this is a file, but this is a analysis that is heavily based on Vijay Nambudiri's analysis in his Nature Neuroscience paper. He's in Stuber Lab for those who aren't familiar with him. And yeah, so the idea, again, in a general sense, is to take those trial averaged event-related responses across all those cells, perform some dimensionality reduction using PCA, and then using clustering techniques, specifically spectral clustering, and there's also a method for doing k-means clustering. They're just different like approaches to clustering data. And then let me just show you how the data look like. So this is just a block for that contains all the packages that we want to load in. I think this should actually run through, because the requirements.txt was created specifically or related to these packages. Does it go through?
I'm getting an error on
The Patsy?
it's the block where we import data and define trial types.
Oh, Okay, so the importing the packages is fine.
Yeah, that one.
Okay, so it might be because I'm wondering if this should be fine. Where are you? So the importing data is where you're having issues.
Yeah, I think it's the 10th or it's right after the Ken analysis one.
Okay, yeah. Let me just quickly see if this runs through for me. I think. Okay, so this is running for me. What does it say exactly? And does everybody else have this issue too?
It just says that there's no file in
Ah, okay.
Yeah, I'm getting the same error. It can't find the pickle file.
Okay. Let's do this. I think.
I even changed the directory so that it was at the directory on my desktop to the file on my desktop, but it still doesn't find it.
Oh, huh. So you did this where you changed the.
Yes. Although, you know what, I wonder, so right now my directory goes to the sample data file. Maybe it needs to go to the next file down, which is the event related analysis file. That's what I'm thinking.
You could try that, but I think as it's set up, should be. Yeah, it's already looking for that event related file. Oh, it could be that. Can you go into your sample data and see if there is an event related dict?
Yeah, mine is there, and I just changed the directory so that it would find that file, and it's still not finding it.
Okay, well, let's do a little bit sleuthing. I think this should be pretty straightforward to figure out. Let me clear this. It works for me. Okay. So do this. Copy and paste this into a new cell. So all I did was Control C, obviously, and then I clicked into the gutter of this block, and then just pressed A, like the key A, and it should make a new cell here. And then I'm gonna do this. And just make sure that this directory is pointing to the right spot. Let's see, superadmin downloads. Does that seem like it's pointing to the right spot? It might be that the error is actually not on this specific line of code.
Yeah, so it can see it now. Do I comment out the line down below then?
Which one?
So I pulled that from the lower. Okay, so you do keep that there.
Yeah, you should keep this because this is just defining the path to this file. Which specific line is it having trouble with? Is it this one?
It says with open data dictionary path RB as pickle handle. It
Okay.
can't find it. So no such file or directory. So even though we found it by adding that new code or that new cell of code, it still can't find it in the next one.
Oh, so this is just like troubleshooting. Actually, this is not actually doing anything. I was just trying to make sure that this is pointing to the right file. That's kind of strange.
Are we able to move that file into like
Yeah, I
the
guess.
sample?
Yes, could do that. How do we do that? Let me see. Yeah, so I think the way that we can do this is cut, or you can copy as well, and then just paste it into here. And then you would need to delete this, I think. So, but we'll try this. I'm not quite sure if that's going to solve things out. I'm wondering if it's,
It solved it for me, but now I have a different issue.
is it here? Is the issue here now?
It's on a different cell now.
Okay, down here somewhere. Okay,
Yeah,
well,
it's on this.
if we run into too many more problems, then I guess I can just run it through and I mean, troubleshooting this is not going to be super important for like for didactic purposes. Let's see. So that still works for me. What's the other issue? Move down here. Maybe I'll run into it. I guess not.
Yeah. It's where we loop through the conditions and plot heat maps to square the heat map.
Okay, so here.
Yeah, and so it has an issue. It's an attribute error on line 19 where it's sns.tsplot population data, bracket sort response.
So this might actually be where things kind of break apart because we didn't have you install the requirements file. That might be like the inconsistencies with the package versions. So I think that's good to know. I'll try to, well, we'll work together offline to get the right packages and get everything running. But I think I'll just kind of explain through the different plots and everything that we'll see. Do things work for you, Jennifer? No?
No, I still can't find the file, but
That's so weird. But the path and everything looks fine. Like it should be pointing to the right one.
I mean, it looks exactly like yours. I'm not sure what.
It has the double slashes as well. It should, like these.
No, it does not have double slashes.
Oh, that could be the reason why. Oh, you know what that could be? When you went here, is there an R in front of the string? That R is actually quite crucial.
Okay. I was going to ask, what is that R for?
Yeah, so.
And yes, it is there.
Oh, it's there. Okay, so then maybe that's not the issue then. I know what it does. I guess in my mind, but maybe explaining it is not quite clear. But it's basically like I guess if you executed this string by itself, these slashes will be rendered as special characters. Like they won't be denoting like divisions in the folders.
Okay, I have it working now. I figured out what was off.
What was the problem?
So on the data dictionary path equals OS dot path join, I had cut out the event related analysis folder and then the pickle file inside of it. So yeah, so if you go down to line 4, essentially in that same window, I had to add event related analysis / event data dictionary. And then it's, yeah, I don't know why.
Like that?
Yep. That's all I had to
Okay.
do. And
That's
now
strange.
it works.
I think that would, okay, that's, I'm confused now, but I'm glad that things are working.
Okay, and now I'm getting the same error.
Oh, huh.
Like, no, is it the Seaborn package? The TS plot?
Yeah, it is.
Yeah.
Okay.
Alright.
Well, I'll figure that out. Okay, I guess for the sake of time, I'll just go ahead and explain these plots and everything. So this is probably a little bit familiar. It's just showing again the trial average responses for each cell in a heat map, and then ROI average as well. Just to kind of show you how there could be some diversity of cell responses. For example, I mean, you might have a cluster here where we have strong activation, and then maybe some cells that have activation for minus, and then some that don't. And then you have maybe a subset of cells where it looks like either it's not really doing anything or having very light suppression, and then not doing much for the minus as well. So those are conditions where you might be wondering, are there kind of different subsets of cell populations that are doing different things during the behavior or the event, and can we in a not, I wouldn't say like not unbiased, but in a not manual way, not human biased manner, can we kind of group those cells together? So I'm going to go through here. So here, this is principal component analysis on the time dimension. So this is just, I mean, we can like talk through this very extensively, but briefly, basically what we're doing here is performing dimensionality reduction on the time domain. Basically collapsing across the time variables, because we have, yeah, let me show you. Where is the data, population data? So we have the data that are being pumped into this dimensionality reduction is ROIs by time, and that time is basically the activity centered around our behavioral events. And typically for machine learning algorithms, they prefer to have fewer of this dimension. So samples or ROIs on the y-axis, and then time or features is what they're called in machine learning, is on the x-axis. And typically the machine learning algorithms want fewer of these of the x-axis variables. And so we do dimensionality reduction to reduce this dimension, essentially. And there's different methods on figuring out how many variables to ultimately keep. For I think that like the default method that VJ had was to find the bend in this plot here, which is basically the amount of variance explained by each principal component, and usually like finding where these two lines bend is I guess an appropriate number of principal components to keep. So that comes out to be 3. Some of these details are, I guess they're nice to know to understand the mechanisms underlying this analysis, but I think for today, it's fine if some of this kind of doesn't make too much sense. Okay, so then we go into the clustering. So we have our our data that we reduced some of the dimensionality for, and now we want to pump the data through the clustering. So if we execute this, what this is doing is different machine learning algorithms have different parameters that you can kind of test out, and those parameters can change how the results kind of look, and they're optimal parameters to that suit to different data sets. So we're making this a little bit easier for you all in that we're basically looping through different combinations of these parameters, as well as looping through either spectral clustering versus k-means clustering. Again, these are just different clustering algorithms. And then we are evaluating each combination of these parameters and figuring out which combination of parameters is producing the best separation of your cells in your clusters. And that's done via this silhouette score, which you'll see that here it's kind of talking about what parameters are being changed or being tested. So number of clusters, and then number of nearest neighbors, and then the resulting silhouette score. And then so ultimately we're going to, let's see, forget where it shows which combination is the best, but ultimately it saves down here the best parameters, and then it performs reclustering with those optimal parameters and the optimal model down here. Oh yeah, here it is. So this is saying like the optimal parameters here. And then this is just visualization of the newly grouped cells. So here you can see that it does, I guess, an okay job at finding this cluster where there's activation in the CS plus and not so much in the minus. This one I think didn't do like the best job, but it picked out some maybe some really like strongly activating cells, and then kind of a mix. And then this one has, I think, more of the suppressing cells. And then I think ultimately it's nice to see, let's see, where'd it go? Yeah, it's nice to see the ROI averaged responses, because I think that the clustering is kind of reflecting these overall trends. So here you can see a cluster that very strongly activates the plus, then one that maybe it's not super significant, and then one that maybe there's some suppression of activity in the CS plus condition. So one thing is that this data set, maybe it's not like the most perfect separation, and that might be, I feel like that's, or my intuition is that it's probably because the low N, like the low cell number. So I think that if you have, if you collate, if you combine neurons from different cell or different sessions and have a larger sample size, these algorithms tend to do a little bit better. And then this is kind of just the separation of those data points in the principal component space. I would disregard this for now because my undergrad is trying to, she has an update that fixes some of this this representation. Yeah, I would ignore this for now. But these are like the most important, I think, plots. Any questions about this? Okay. And then finally, I think another analysis that might be people might be interested in is, I'll just run through this quickly. Here, this is plotting the like a mean image of your video, and then also plotting the contours of all the ROIs that you have, and coloring them based on how active each cell was for a given condition. I think I already have this evaluated. So the plots that are important are down here, where you can see that we have our ROIs that we had already drawn, and the color indicates how strongly the cells activated for each event. So we can kind of see how that's where the color is extracted from here, where these are trial average responses that came from the event-related analysis code. And you can see that this particular cell activates very strongly to this is for the CS minus. And so accordingly, that cell 1 is colored in brown. And then all these other lightly colored ones are likely low activation or suppressing activity. And then we can also do that for the CS plus condition. And you can see that there are potentially different distribution of cells that show different response patterns. And again, deriving it out off of this this trial average response. And then finally, this is a bar plot where you see each ROI on the x-axis, and then the time average responses, time and trial average responses for either the CS minus on the left or CS plus on the right. And this is kind of gives a summary of how the different cells can respond, and you can compare the response magnitude between the different conditions that you have. So maybe, like for example, this, you can see that for the most part, these cells, the ones that have strong responses, prefer the CS plus, which I guess kind of makes sense given what we've seen already. But I think maybe you'll see different response profiles for different behaviors that you can put in. Okay, so yeah, these are the post-processing pipeline scripts. Any other questions?
Charles, does the plot activity contours, does that work if you run the data through Suite 2P?
I need to test that out still, but just thinking about it, I think,
because it's looking for the similar
right, yeah, it is
path.
looking, yeah, you're right. So this is, yeah, this is currently working, I think, only for the NAPECA pipeline, but I can imagine that I think it's probably, is this analysis that you want to run like kind of soon?
You know, it is probably the most visual
Yeah.
and intuitive for an audience.
Yeah.
Yeah, so it would be good to get this soon. I mean, not like tomorrow,
Right.
but yeah, I'm interested in getting this running for Suite 2P. Absolutely.
Okay. Yeah, so I mean, that's definitely like the goal of everything. So I can make that a priority. I think there's other code here as well to just like plot the contours just by themselves. And I have another piece of code that basically plots these so all the ROIs are not just green, but different colors. And then you can plot like a line plot that shows the activity across the whole session for each cell with the corresponding colors that the contours are.
Is that the analysis that you sent me the other day?
Yeah, that's from that's the code that generated the plots that I sent you the other day.
Yeah, that's fantastic. It was very, very good data.
Yeah, so I think that needs a little bit more cleaning up too, but I can focus on that. This week I have a little bit more time than last week, so I can work on that.
Well, do let me know how I can help, because I'm very interested.
Yeah.
This was super helpful.
Yeah. I hope it's helpful in that it kind of can serve as a springboard for you all to just get a general sense of how the data look. I think I'll admit that I think in the current state, it might be a little bit confusing on how to adapt your data and load it in. But I think part of the reason why that I wanted to do this meeting was also to get a sense of what people's data look like, and how I can spend the next few days to write up some scripts to adapt those data to seamlessly load into these files. So I think that's just the missing link. Otherwise, I think this video that we've just recorded into the session, aside from like I guess a few of those bugs which I'll sort out, once you have those converted data, you should be able to like run through most of these scripts. But I think to move forward, how about let's see. So Victoria, I think. Well, I already have the from Meson, like in Scopix sample data extracted time series file. So I could work with that, or if you want to send me one of your Excel sheets or the CSVs, that could be helpful too.
Yeah, I can do that.
Yeah, and then Jennifer, Sweet 2P, I already have, I mean, I can generate those data already. So your event data.
Yeah, I mean, the event data for mine is difficult, right? Because my event is a continuous measure. Yeah. So my thought right now, because we're using that cross-correlogram analysis in order to align activity and circulating glucose, I think that's the pipeline I'm going to have to get up and running. And I already have all of that code. I just need to figure out how to load essentially all the individual cells' CS files into it. So I don't think it'll be too bad.
What do you mean by load individual cells?
So instead of so right now the code loads individual photometry experiments
I see.
by the entire group. And so I just need to adapt that so that it's pulling in individual cell files, individual ROIs.
Yeah.
Yeah.
I think that should be pretty straightforward. And you can kind of look through this code to see how I do that if you want.
Mm-hmm. Yeah, no, I don't think it'll be hard at all.
Yeah. Okay. So yeah, I'll work on all that file like transformation stuff for both Scopix and Sweet 2P.
Well, I can't thank you enough. This is really a wonderful, really wonderful introduction.
Yeah, no problem.
Yeah.
My pleasure. Okay, so we can end it there if you all don't have any more questions. For the errors that came up, could you like screenshot them and just send them to me, and then I'll sort through them.
Yes, I can send that one I took a screenshot of and I'll send it to you now.
Okay, great.
I can send the one that I got.
Okay. And then I guess we could work together sometime soon to get Anaconda working for you.
Yeah, thank you.
Yeah, but it
I can
seems
also
like,
just like search around on Google and
Okay,
try to
sure.
figure it out. Yeah.
sure. Well, just let me know if you run into any like roadblocks or anything.
Okay. Thank you so much,
Okay. All right. Hopefully everyone has a good time on analyzing their data, and talk to you next time.
Thank you.
Thanks,
Yep.
Dan.
Thank you so much.
Bye.
This is super helpful.
Yeah. See ya.